Okay, so today we will continue with the complement system again. Yesterday we had done the classical pathway, and today we will study on the alternative pathway. Okay, so let's see how the alternative pathway works. Okay, so this is alternative pathway. Now coming to alternative pathway, why has it been named as alternative pathway? Because it was discovered after classical pathway. Okay, so it was an alternate method that was found out, and that is why it is known as an alternative pathway. This was found that the first response of the innate immunity or innate immune system is the alternate pathway. Fine. Now, going forward, we had seen about the different proteins that are present in the complement system. Then out of the many proteins, the C3 protein is the most abundant complement protein that is found in the body of of the host. Okay, now in alternative pathway, what happens? This C3, they undergoes through a spontaneous hydrolysis. So that means hydrolysis. What hydrolysis means? Breakdown of water molecule. So here the C3 undergoes hydrolysis that leads to a formation of the new compound which is known as C3 water. C3H2O is the compound that is formed because of hydrolysis of C3. Okay, now this C3 water, what happens as it is formed, as the C3 is cleaved or as the hydrolysis of C3 happens, it opens up a receptor area for this B protein. Now the next component which is coming towards and binding to C3 water is protein B protein or B complement. Okay, so the B complement will bind to C3 water. So after this factor B binds to C3 water, what will happen? Another factor called B will move forward and it will cleave this factor B. Now factor B, after cleaving, it breaks into two parts. One is BB, capital B and small b, another is capital B and small a. The capital B and small b will remain attached to C3 water while BA will be released. It will it will move away. Okay, so this capital B small b is a larger fragment and this is a smaller fragment, and this one is released and it goes away from here. Okay, the next, this whole complex which is formed of C3 water and BB, this has got protease activity. That means it has the activity to lyse protein. So what happens when C3 protein comes and binds to this particular complex? It will now be cleaved. The C3 protein will be cleaved into C3b and C3a. Okay. Similarly, if more and more C3, since C3 is present in abundance in the host, for more and more C3 will come and more C3 will be cleaved, and this is how they are separated. The C3b fragment will be larger in size and C3a will be smaller. Okay, so this whole protease is also known as C3 convertase because it has the ability to cleave C3 into C3b and C3a. So this is a convertase. This is known as C3 convertase, which has the protease activity. Now those C3b that were formed from C3 after C3 was cleaved. Now few of the C3b, what they do, they go and they get attached to the microbial surface. Okay, this is a microbial surface on which it has attached itself. It has fixed itself. So this is known as complement fixation. This is C3b is a complement. So this complement has fixed itself. So the complement fixations occur. That means this C3b covalently binds to the microbial surface. Next, what will happen? Again, as where we have seen, the B factor will come and join to it. Right? Then after the B factor comes and join to it, the D factor will come and cleave B into BB and BA. BB will remain attached to C3b and BA will be released. Okay. Then when the C3b capital B small b complex is formed. This is known as C3 convertase. So similarly, again, more and more C3 will come and it will like start to bind with it. But prior to that, what will happen? When the C3b and BB complex forms here on the microbial surface, it is not that firm, it is unstable. So to provide stability, another component which is known as properdin, which is designated with capital P, it comes and joins to it to stabilize the complex. Okay, so properdin P stabilizes the C3b BB complex. That is its role. Then what will happen next? Until now, what we have seen, C3 hydrolysis has occurred, formed C3 water, on which B factor had come and joined. Then D came and joined, and then it cleaved to B into BB and BA. Then this BA was released, and BB remained attached to C3 water, and then this forms the C3 convertase. Then whenever C3 came and joined to this convertase, it was cleaved into C3b and C3a. Then sometimes C3b goes and attaches itself on the microbial surface. Then what happens as it attaches itself to the microbial surface? Then the same process will repeat here. Okay, the C3 on C3b, now the B factor will come and join, and to it the D factor will join and cleave it into BB and BA, and then it will form a this one convertase again, that is C3b BB, and since this is unstable, properdin will come and stabilize it by forming a covalent bond. This alternative, this C3b-BB complex is known as the alternative C3 convertase. Okay, so this is what we have studied until now. Then, what is its role after that? Okay, let's see. Did you all understand this part? Okay. So see what's happening here. This is the C3 protein that was present in the host. Then it was what did happen? It was hydrolysis occurred, and then it formed a compound which is C3 water. Now factor B is moving towards it and binding to it. Okay. Then next step that takes place is a factor which is called factor D will come, FD in short, will come and bind to it, and it will cleave B into BA and BB. Then factor D goes away and BA is released, and it just forms the component called C3 water BB. Now C3 water BB is what? This is the C3 convertase. Now what will happen? Alternative pathway C3 initiation convertase is formed. Okay, so the C3 will be alternative pathway will be initiated now with this help of the C3 convertase. So when a C3 comes and binds to it, it is again cleaved into C3b and C3a. Now C3b will move and it will attach itself on the microbial surface. So this blue color thing you see below is the microbial surface on which the C3b has bound itself. Okay, it has attached itself. So that is complement fixation has occurred. Then again the same process will move on. Factor B will come and join to C3b. Factor D will join to factor B. Then it will cleave factor B into two parts, which forms BB and BA. C3b and BB will form the C3 convertase. Now, whenever more C3 molecules will come and bind to it, it will start cleaving. Okay, now you see how more and more C3 is coming forward and binding itself trying to bind itself to BB, and it is getting cleaved. Okay, so this will continue. See, more and more they are coming and binding and they are cleaving. So at one point of time, some C3b molecules will move away somewhere. Some will again sit on or attach themselves on the same microbial surface. So this is going to continue. So this way, and some are going to bind itself on the C3b or C3 convertase itself. Okay. So when this binds itself to C3 convertase, once it binds itself to C3 convertase, that becomes, see here you see that C3b, one C3b part has come and bound itself to the C3 convertase. When this binds to C3 convertase, this forms another convertase which is known as C5 convertase. Okay, now what will happen? Any C5 component will come and protein will bind. The C5 will be now cleaved. C5b remains attached and C5a goes away or it is released. So there we see C3a and C5a both are separate and floating, and they have the same property which we had seen in the previous pathway, in classical pathway, that is chemoattractant. They work as chemoattractant. They will attract the basophils and the mast cells. They will attach to them and then they will activate those mast cells and basophils to release the granules, and then they will help in killing the or destroying the cells. Okay, that was the role of C3a, and similarly C5a also does the same thing. They will attach themselves to this macrophages and neutrophils, and then they will again like use them in destroying the cells. So this is what they do after getting released. And coming back to again our previous thing, that was the C5 convertase that was formed on the microbial cell. Now from that, the C5 convertase is already there. So few of the C5b molecule, they go ahead and they form a separate area for themselves. Now this C5b, there it has separate, instead of going and attaching itself to the convertase, it has moved forward and then it has grown larger in size, and then it's trying to now form something here. Similarly, again C6 will come and bind to C5b, C7 will come, C8 will come. Okay, and then C8 starts preparing up, making a pore over here. C9 in large numbers accumulates in this area, and then the pore enlarged. Okay, and what happens finally? All kinds of water and other electrolytes and all enters into the microbial cell. So this is what is it forming? Membrane attack complex. This is forming a complex which is attacking the membrane. Okay, so this is how the alternative pathway works. The third pathway that we need to study in the complement system is the lectin pathway. Okay. Now this lectin pathway, in the lectin pathway, there are some proteins which are known as lectins. Okay, what is the property of these lectins? The lectins are proteins which are soluble. Okay, they recognize specific carbohydrate components which are present on the microbial surfaces. Now, what will happen in this particular pathway? The lectin which binds to the mannose, that is known as mannose binding lectin, was the first lectin that was to be discovered. Okay, this mannose binding lectin, it looks like this. It's known as MBL. So this MBL, it binds to mannose containing carbohydrate. Those carbohydrates which contains mannose. Okay, that means this MBL, mannose binding lectin, or you can say in order to activate the lectin pathway, when will it activate? It will get activated only when the bacteria, fungi, protozoa, and virus which enters, they have components or they have carbohydrates which has the presence of mannose in it. If the mannose is present, then this mannose binding lectin will get activated and those MBL will bind to the mannose. So that is when this lectin pathway gets activated. Okay, then this MBL, mannose binding lectin, where is it present or where does it circulate? It circulates in plasma. How? As a complex with two proteases. Two kinds of proteases are there. One protease is known as MASP1. Another protease is known as MASP2. So in the presence of these two proteases, MBL circulates in plasma. Okay. Now, what are this MASP1 and MASP2? See, if this is an MBL, this part is known as, you will see two structures attached to MBL. The structure on the left side is known as MASP1. The other one on the right side is known as MASP2. What is the full form of this? M stands for MBL, A stands for associated, S for serine, and P for protease. MBL associated serine protease. So these two will be attached. Now let's see how it works. So this is a microbial surface. So on this microbial surface, for example, there is a presence of mannose, and that is why this MBL complex bound itself on the mannose which are present on the microbial surface. Okay, and it has those two things on its side, 1 and 2. Protease MASP2 here, this one. This one acts as the convertase, means the role of cleaving will be done by this MASP2. Okay. So first of all, C4 protein comes, it's come towards it. Then C4 protein is hydrolyzed, not hydrolyzed, relieved by this MASP2. C4b and C4a are formed. C4b is smaller in size, bigger in size, and it attaches itself on the microbial surface. And C4a is released. Okay. Then again, the next protein, that is C2. C2 comes forward, and then it tries to bind again, and it is again cleaved by MASP2 and forms C2a and C2b. This time the C2b is released out because C2b is smaller in size, and C2a, it binds itself to C4b. And now these two remains attached. The C2a and C4b remains attached to on the microbial surface. After this forms a complex which is known as C4b C2a complex. Okay. Now what happens to this complex? This complex forms C3 convertase. Okay. So in short, again a revision of the part which I have told you just now. Binding of MBL to microbial surface activates lectin pathway. So MBL has bound itself. MASP2, this part has the protease activity. So first of all, C4 comes and tries to bind, and C4 gets cleaved into C4A and C4B. C4A gets released, then C4B is remain attached on the microbial surface. Then again C2 comes and C2 is cleaved again by MASP2. C2B this time gets released, and C2A remains attached to C4B. This altogether, C4B, C2A forms a complex which is known as C3 convertase. Okay. So this is what happens. And after that, what happens? Now we have stopped till here. Next, what will happen? This C3 convertase which forms will start cleaving C3 protein whenever it comes to it. Then it will continue the same way how we had studied the alternative pathway. C3 will be cleaved, C3b will be formed, C3b will go and attach itself to the microbial surface, and then so on, it will go on like forming more and more convertase, and then at last it will form the MBL, and then the whole cell will be destroyed. Is this pathway clear to you all? We have finished with all the three pathways of the complement system: the classical pathway, the alternative pathway, and the lectin pathway. All the three of them at last ends up with the same thing. So they are all interrelated. You can, okay, then we'll stop the class here for today. Meet you in the next class.
Thank you, ma'am.
Yeah.
Thank you, ma'am.